In this video, I will be showing you how to do the spread plate method, but before we get started, you need to clean up your workspace. Use germicide to wash down your workspace and then use paper towels to wipe it up. Then, after that, you may gather all the materials that you will need for this experiment. For this experiment, you will need gloves, paper towels, germicide, goggles, plastic petri dish, which will stand in as your agar plate, lab coat, China marker, cell spreader, 95% ethanol in glass petri dish, biohazardous waste, plastic pipette dropper in place of the micro pipetter, a Bunsen burner, and liquid stock culture. Now for the real experiment. To start, label the bottom of the agar plate. We don't actually have one, so we're going to use a plastic petri dish. And what you're going to do is put your initials and the microbial identification on the bottom of the petri dish. Like so. Then sterilize your cell spreader. In order to do this correctly, you want to first douse the cell spreader in ethanol. Then briefly pass it through the flame of a Bunsen burner. You want to allow the ethanol to burn off. You do not want to hold the spreader in the flame though. Now I will be using my plastic pipette dropper. I will be transferring my liquid stock onto the surface of the agar plate. In a real experiment, I would use a sterile micropipetter to transfer my liquid culture, but for this video, we're just going to use this plastic pipette dropper. And yes, in a real experiment, my test tube would come off the test tube rack, but I didn't have one, so I just kind of found whatever was lying around my room to hold it. So, yeah. So what you're going to do is take the cap off the test tube, get my culture, and what you want to do is get 0.1 milliliters of this liquid culture. Put the cap back on and then put the test tube back on the test tube rack. Then I'm going to be putting my liquid culture onto the surface of the agar plate. You're going to open it using the clamshell method and then putting the culture in it. Then close the lid, and then dispose of my pipette. In a real experiment, I would dispose of it in the biohazardous waste container, but I don't have one, so I'm just going to put it on my desk. Next, I'm going to sterilize my cell spreader again. First, I will be dipping it in the plastic petri dish filled with 95% ethanol. And then I will pass the cell spreader briefly through the flame to burn off the alcohol. Then spread the specimen over the entire surface of the agar using the cell spreader. First, you're going to open the lid like a clamshell, then gently push the cell spreader back and forth across the specimen. You want to continue to spread the liquid until it forms an even layer on the surface. You can spin the plate to get an even spread. Kind of want to get it in every inch of the plate. And when you're done, just put the lid back on. Then sterilize the cell spreader again one last time. Once you've done that, put it down on the bench top. Now, let this plate stand for 5 minutes before inverting for incubation. After this spread plate is ready to be incubated, incubation is for 24 to 48 hours and at 37 degrees Celsius. After that, you are now ready to decontaminate yourself and your workspace. First, dispose of your gloves in the biohazardous waste container. Your gloves are now contaminated from the experiment that you have just conducted. After you've done that, wash your hands thoroughly. Then use germicide to wash down your workspace, and then use paper towels just to wipe it up and dry it off.